So we saw the basic idea about how some of these genetic tests can work. Let's take a look at immunological type methods. And with this, we're going to be talking about antibody-antigen complexes, or how antibodies and antigens are going to help us figure out results for these tests. So here we're going to look at serology, so that is the utilization of serum to run these tests. And within the patient's serum is where we're going to find these antibodies. Okay, remember the difference between plasma and serum. Plasma is going to have those clotting factors, where serum doesn't. So when we're running these tests utilizing serum, a lot of what we're looking for are going to be clumping results. You don't want to have those clotting factors present. That's why we use the serum. Okay? So when these are run, you either need to know what the antibody is, or you need to know what the antigen is. So you need to know one or the two so you can figure out the other one. So if you know the antigen, you're trying to figure out the antibody is. If you know the antibody, you're trying to figure out if the antigen is there, okay, or vice versa. Okay, so it's all based on that idea that antigens and antibodies are going to be specific. That antibody is specific for that antigen. Okay, remember the antigen, that is that foreign entity. The antibody is what you are going to produce in response to that antigen. Okay, and oftentimes you see this based on some type of a clumping, a precipitate, an agglutination, something like that. So yet again, okay, an unknown antibody can be detected as long as you know the antigen, and vice versa. An unknown antigen can be detected as long as you know the antibody. You have to know one or the other. Okay, so you can take a number of different samples. When we run this, okay, depending on if you're looking for antigen or antibody, of course. Okay, antibodies would be in the serum. So here, let's take this picture right out of your book. It is awesome, and it'll help to illustrate this idea. But there's two different things going on. So let's just look at just this left side first. So here's our question. Does the patient make antibodies towards the microbe? Use the example of like an HIV test. Does the patient make HIV antibodies against HIV? If a patient does, that is because they are exposed to that virus. They have that virus. They have that antigen present. If they don't have HIV antibodies, it's because they don't have it present. Okay? That's what we're trying to figure out. So does the patient make these antibodies? So you take the patient's serum. You don't know what antibodies are there. Okay, so remember, you have to know either the antigen or the antibody. So in the serum, obviously you don't know what's there. Okay, you don't know which antibodies are present. So then you take the known antigen. So HIV proteins, let's say. Here they are. Mix them together. If there is clumping as a result, it's because antibody attached to the antigen, and you see that visual result. If they're not specific for each other, then you don't see that clumping. Okay, this is just the premise, just the idea behind what we're looking for. The only way you should see a positive result is if that antibody is there. Okay. On the other side, it's the same idea, just flip-flopped. Okay. What is the identity of the microbe? Which organism is infecting the patient? The question is, does the patient have a Staphylococcus aureus infection? Yes or no? So here's what we do. You isolate the organism from the patient. So you have the organism on a plate, okay, and you're like, is it staph? How can we figure out if it's staph? Well, here you take anti-serum, okay, which are the antibodies in the serum. So you take the anti-serum for staph. So these are antibodies for staph. And you're going to combine them with the antigen, the foreign entity, the Staphylococcus. And you look for the same thing. If you see the clumping, then you can say, yes, this unknown organism is staph because it clumped with the antibodies. If there is no clumping, you can say, okay, this unknown entity, is not staph aureus because it's not attached to it, or the antibody is not attached to it. Again, this is the theory behind this. We're going to explore tests which utilize this theory. Okay, so that's what we're looking at. So this picture is fantastic, just to get you to understand the concept. Okay, and the concept, again, antigen, antibody are specific to each other. You need to know one or the other to determine the other one.
Can there ever be a false positive on those?
Yes. And actually, that's where we're going to go right now.
Oh, okay, good. That's what she's getting at.
Exactly. Yeah. Okay. So when it comes to running different tests, we have what's known as specificity and we have sensitivity. This is going to get a little confusing, okay? You need to keep straight the difference between specificity and sensitivity. If we just think about what specificity means, it means how specific something is. If we think about sensitivity, we're thinking about how sensitive a test is. If it's able to detect the smallest amount, then it's sensitive. If it needs a lot, it's not as sensitive. Or if it can read a lot. So specificity, here we're looking for how specific it is. So if you have a very specific test, it only focuses on that antibody, okay, a specific one, or maybe an antigen, whichever. Very, very specific, right? It will not react with anything else. Here's what we need to remember. There are not a lot of tests out there. It's really hard to find something which is very specific and very sensitive. Really hard to do. Most of the time, as specificity goes up, sensitivity goes down. Or as sensitivity goes up, specificity goes down. It's usually like this, which is a problem for a number of tests, just how it is. There's a few which are on the higher side, and we'll talk about one of them today. I'll go through steps with that one today. But looking at this, okay, specificity is just referring to the ability to react with what it needs to react with only. So if we look at this little example, this picture is right out of your book, okay, specificity example. Up here we can see sacrifices sensitivity. So it's very specific, it's not as sensitive most of the time. Okay, so here we see the antibodies. The Y-shaped structures are the antibodies, and they are made. They are going to react with this specific antigen. Okay, so with this red-colored antigen, diamond-shaped antigen only. We can see that there's other ones that it's not reacting with. We can also see that there are some that it hasn't, that it should be reacting with, but it really hasn't. Okay, it's very specific, so it's made to react with just that antigen. Now, if it's very specific, it's not as sensitive. So what that means is there has to be a lot of these guys around before they'll pick up on it. That could be a problem with a lot of tests. A very popular test, which is very highly specific, but not very sensitive, is your rapid strep test. Go in, take a, let's say you're going in with a sore throat, whatever, strep throat. This was like, well, let's test for strep. They'll run that rapid strep test. Okay, a lot of the time, they come back and what do they say?
Negative.
It's negative, right? And then what do they say?
They're culture.
They're in a culture. Okay. And we know this. Okay. Why is it negative? Okay, because it's very, very specific. Basically, there has to be a lot of antigens. There has to be very heavy growth of that organism there before the test will pick up on it. Why is that test so specific? Because it doesn't want to pick up on anything else similar. It doesn't want to pick up on anything else that's going to be present in the throat. Only wants to pick up on strep. But because it is so specific, its sensitivity is lacking. So that means there has to be a lot of it. So earlier stages of strep throat is probably going to come back negative because there's not enough yet. Sure, later on, it's really, really bad. Chance of that coming back positive is going to be much greater because there's going to be a lot of antigen around. Okay, so think of that. Rapid strep test is an awesome example of something very specific, but it's not very sensitive, meaning there has to be a lot there. and run it for a culture and see if that comes back positive. See if we get some growth. Now with sensitivity, this is made to detect even the smallest amount that might be there. But what happens when sensitivity goes up? Specificity typically goes down. So it's not as specific, but it's very, very sensitive. So it'll pick up on the smallest amount. So if you think of it like an HIV test, think of an HIV test, you want to be able to pick up on the smallest amount of antigen there, right? Okay, that would be ideal. But the problem is, because it wants to pick up on the smallest amount, you lose that specificity, so it might pick up on things that it shouldn't. So in this scenario, you would see a number of false positives, which, if you're looking at HIV, false negative or false positive, in this scenario, you'd rather have the false positives because that is definitely going to ensure more testing, to confirm that. You don't want someone leaving thinking they're negative and then spreading that infection. Okay, so this is the exact opposite scenario of what we saw with the strep test. So if we look at this picture, okay, so highly sensitive. So pick up on the smallest amounts present. So the purple antibodies, they are picking up on the purple circular antigens. And you can see there's just a few of them. In this picture, there's 5 out of all of these antigens. Okay, so it's made to pick up on the smallest amounts, but again, it's not very specific. So look, it picked up on this yellow one. It picked up on one that it shouldn't. That's the problem. If it's very sensitive, a lot of times that specificity is lacking. Picks up on things it shouldn't, and then the patient gets a false positive. Okay, so definitely I want you to understand how the false positives or how the false negatives can happen and what sensitivity and specificity refers to. Okay. Agglutination and precipitation, with these types of reactions, they're basically the same thing. There's like one big difference between. Depends if you're looking at a whole cell or a soluble molecule. Okay. So if you look at agglutination, the antigens are a whole cell. With precipitation reaction, the antigen is some type of a soluble molecule. It's not an entire cell. Is everything else the same? Yes. How are the results visualized? As some sort of clumping, as some sort of coagulation, some sort of, think of chemistry where you did a precipitation reaction. There's some type of precipitate, there's some evidence, that there's some type of blob or clump or precipitate, something is formed. Think of when you've done blood typing in the past, you're looking for that clumping, that agglutination. Okay? That's a positive result. The only difference between the technical terms agglutination and precipitation is what the antigen is. If it's a whole cell, it's agglutination reaction. Soluble molecule, it's precipitation. That is it. Simple as that. Okay, both reactions, you're going to see that you're going to have some type of a precipitate, some type of a clump. Something settles out of solution. You see that clump. Okay, agglutination versus precipitation, again, you see difference, whole cells with agglutination. Okay, cell-free system, just molecules with precipitation. That is it. Okay, blood typing, if you've done blood typing, okay, again, that's agglutination. You're using the entire red blood cell. Okay, detecting those antigens on that red blood cell. Precipitation, it's a cell-free system, RPR, rapid plasma reactant, I think is what it is. It's a syphilis type of test, which would be an example of a precipitation. Agglutination, again, you're using a whole cell. Okay? A cell system. Okay? Clumps of whole cells with agglutination. Blood typing, fantastic example. So looking here, we can see here they're using like a micro titer plate, to do this blood typing. You see the agglutination, okay, versus non-agglutination. So you can see the clumping with agglutination. Over here, we're looking at an antibody titer. Okay, an antibody titer we're looking at the concentration of antibodies, okay, within that patient's serum. Typically, the higher that antibody titer is, the higher the concentration of antibodies, that's going to confer greater protection for that patient against that said antigen. Okay, so the titer is highest dilution of sample. You see that reaction, that's going to give you the concentration of antibodies. So here, they're running a series of dilutions. We can see it's going to be diluted from 120 dilution down to a 140. So we're diluting it, and we're still seeing that clumping, that agglutination. So you're going to want to go until you don't see it anymore. And it's the last one where we see that clumping. Okay, so that titer would be 160. Okay, so that's the concentration of the antibody within that sample. So another example of agglutination, and then precipitation. Think of chemistry, just look for that precipitate. Again, difference is not a whole cell, you're just looking at a cell-free system. So you'll see something like this. That would be your precipitate. Here are those soluble antigens, and again, antigens are a soluble molecule, not an entire cell. Here are the antibodies. If they attach, you see that precipitate. That is it. Okay. Very simple difference between the two. So we need to go through the Western blot and then the ELISA, and that's our lecture. And again, the ELISA is what we're going to do on Tuesday, the HIV ELISA. First, we need to talk about Western blotting. Western blotting and ELISA, they're a little bit complicated. They're similar, they kind of have some similar principles to them. I definitely want you guys to understand the order of these steps and why they're happening. Something I want to put up here before we get started. There's different types of blots. There's Western. They're named after like every direction. There's Western, there's Southern. There's Northern and there's Eastern. And they each measure something different. They each use something different. So for Western blotting, this first bullet point up here, we can see there's a sample of known proteins. Okay, so Western blotting is going to use proteins. So with HIV proteins. Southern blotting uses DNA. Northern blotting uses RNA. Eastern blotting uses carbos or carbohydrates or lipids and so on. Western blotting is very, very common, as in Southern. Okay, so the Western blotting, not the easiest test to run. There's a lot of different steps with it. So when we look at this, the first thing you need to do is you need to separate out those proteins. So when you were doing your research, maybe you came across something that looks like a well, that looks like this. A gel, pardon me, that has all those wells in it, and then maybe you saw bands at different places. So, like that, okay. Now, and you can see one. I'll explain this in just a moment, but if we look here,
I had to a little bit.
we can see one here. So basically what happens is you have to make this gel, right? It's kind of like making Jell-O. You pour it into this container, you insert a comb, you let it solidify, you pull the comb out, and then you have these little open wells. What you do is use a little pipetter, you draw up your proteins, and you put them in each well. Great, so now we have the proteins in that well. You have to hook it up to an electrical charge. If we're looking at organisms, we have the negative charge associated with them, so you have a positive and negative charge, and they're gonna run down the well based on electrical charge. The larger the protein is, means it's going to get trapped in the beginning of the well. The smaller ones can go further and further and further through that gel well, basically. So the smaller proteins make it to the end, bigger proteins get trapped at the top. Important to realize that. Okay. So they're distributed throughout the gel, and then they're transferred and put on a filter. The way we do this is very different. It's just kind of depending. I remember when I did it back when I was in grad school, when I had to run these, we actually had to use filter paper and paper towels when we're taking textbooks and stacking them on top so it can effectively blot that filter paper. But basically you want to get evidence of these proteins onto this filter. So at this point, you have this filter. You have those proteins on this filter, okay, spread out how they were in this gel. Now what you need to do is you add the patient's serum. What's in their serum? Antibodies. On that filter, you have the antigen, the foreign proteins. You add the antibody. What you're looking for is if the antibodies are there, they will attach to the proteins. That's what we're looking for. Okay, so if the serum contains antibodies to that microbe, they'll bind to the antigens on the filter paper. They'll actually attach. But you can't see that, right? You can't see antibodies with the naked eye. How do you even know that they attached? This is where it gets complicated. I mean, so far, all you did was spread out the proteins, got it on a filter, okay, antigens are on the filter, add this liquid, serum that contains antibodies. They may or may not attach, but we need to see if they attached. So, next up, you have to add a second antibody. Here's the deal. That second antibody has a little fluorescent tag on it. It is made to attach to that first antibody. So if that antibody is attached, the second one binds to it, the tag will glow. If no antibodies are attached, the second one has nothing to hook onto, it will not glow. Simple as that. It's the only way you can effectively see the results. And the results, will just be the bands, like this. Okay, let me see. Okay, so again, you take those antigens, you separate them out on that gel, transfer them onto a filter, add the patient's serum containing the antibodies. We'll see if they attach or not attach, which you cannot visualize. So next up, you have to add that second antibody, which is made to attach to any antibody, which is lingering, which is stuck to a protein. And then it will glow. You add some dye and look at it, it will glow. Okay, very specific and sensitive. So Western blot's a really good test to run. A common HIV screening test would be ELISA. Would be the one we're going to run in lab. This one, you can get some false positives with it. So what they can do, Western blot, next, it's highly specific and sensitive. So it's a good confirmatory test for this ELISA. Okay, so Western blot, you guys should know, is oftentimes utilized as a confirmatory test for HIV. Okay, so Western blot is used as this verification confirmatory test for HIV. Really good test. Here's an example of results. Looking at different proteins, you can see the numbers get smaller as you go down because the proteins, are numbered based on size. So they're smaller as we go down. Glycoprotein 160, protein 55, protein 24. Here you see the bands, what they have. And this picture is really cool because it shows exactly what we did. Okay, here's your filter paper. Your blot. Remember you blotted whatever was on that gel, you blotted it onto the filter paper. And again, the protein, the antigen, is blotted on that filter paper. Next up, You add the patient's serum. Maybe the HIV antibody is there, maybe it's not. In this picture, it's there. So it attached to the antigen. Next up, you have to add that second antibody. It's the only, because this we can't see. So we have to add the second antibody that has a little dye tag. And then if this one is here, second one attaches, it glows, that's how you know it's there. It's there, it's there, it's there, it's there. Okay, so you can see that band.
Lori, I'm confused.
Sure, what's up?
If you, I think I'm missing something. If you're blotting it onto paper, then why is it showing up in your gel? Why is the fluorescent?
This isn't the actual gel. This is not the physical gel itself.
So it's not the gel?
No.
What's that?
That's just the paper.
Oh, okay, now I got confused.
Okay, good. There's other tests that you can run where you're staying with the gel.
Yeah,
The blotting type of test, it gets blotted on the filter paper.
okay.
And then this is showing what you see
So
on the
the
paper.
glowing comes on the paper?
Yes, yes. You just soak the paper in some solution and then they'll pop. Okay, so here we can see evidence of an actual Western blot test for HIV. So this is a particular patient. So if we look at this, we can see different bands. So first up, down here, so here we have just our source band right here. This is like our positive control, where you should see a positive result of this glowing for every single protein all the way down the list. Okay, you should see. The next one is your negative control. Okay, so your negative control should only glow just for the patient's serum. Okay, so serum control only, no other bands. So positive control, negative control, you run those with any test that you run. And then patient samples over time. See if these bands appear. So here we can see which proteins are present. So first up, in order for a patient to be positive, they have to meet two different conditions. Okay. They have to be positive for GP160, glycoprotein 160, or glycoprotein 120. They have to be positive for one of the two, and positive for one of the two other ones. So looking at this, GP160 or GP120, is this patient positive for one of the two? Absolutely. Here we see GP160, pretty evident. It looks a little bit like GP120 is starting to appear. Okay. Now, the other set of conditions is they would have to be positive for P31 or P24. Okay, so we look at that. P24, very, very apparent. So if they have one of these two is positive and one of these two is positive, that would be a positive diagnosis. So this patient, would they be positive or negative? They would be positive. And then on the Prezi, I have those standards down here. Okay, so they're positive if GP120 or 160 is positive, and P24 or P31 is positive. And this is right out of your book. Okay, I took the sample from your test, so it's in there.
Is that just for HIV?
Yes,
Okay.
HIV specific proteins. So that's the Western blot. Last thing I have is this guy. Last thing I have with lecture is looking at the ELISA. So this is what we are going to run on Tuesday. So it's really nice for getting a good look at the theory behind this before we actually run it. So let's talk about how this works. You don't have to have your lab on by any means. I'm just going to cover everything up here. Basically, we'll cover it by looking at a picture, which is right out of your textbook. So whenever we run an ELISA test, first off, you guys need to know what it stands for. Okay. You refer to it as ELISA. It's enzyme linked. So know there's going to be an enzyme somewhere in there. Immunosorbent. Okay. Sorbent means something attaches. Okay. Immunosorbent, so immuno, here we're looking at an immunological type test, antibody antigen type test. Assay means use small quantities, assay type test. And we're going to use little tiny microtiter plates. This picture down here This is a microtiter ELISA plate with 96 tests, 96 wells. I think ours, we have them big, but our lab tech will cut them down. I think we're going to have 12 wells per group. Okay, so usually they're strong. I have one down in the lab to bring it in this room. But usually there's hard plastic type of material. And you can see any color change is going to indicate a positive result. Okay, if it stays clear, that would be negative. This is an indirect ELISA. There's indirect and there's direct. Indirect, you're looking for the antibody. We are going to run an indirect ELISA. More common. Okay, indirect ELISA looks for the antibody in the patient's serum. A direct ELISA, you're looking for the actual antigen, the foreign entity. Okay, so you're directly looking for the antigen with the direct ELISA. Indirect, you're looking to see if they're infected based on the idea if they produce the antibody. So that's why it's indirect. You're not looking for the agent, you're looking for what they did in response to it. So indirect ELISA, you're testing for antibodies. So in the case of HIV, do they have the HIV antibodies? Okay, so you need that solid support, like that microtiter plate, because these molecules need to stick to the well. They need to stick to the sides of the well. All right, so we're just going to run through these steps. You'll see a few similarities to the Western blot as far as, antigen-antibody affinity for each other. So indirect ELISA, testing for antibody, again. So what we're going to do is we're going to look at a positive reaction versus a negative reaction. Okay, so this would be what we're going to see in a patient's well that's positive, what we're going to see in a patient's well that's negative. See the difference. And again, with your naked eye, you're not going to be able to see anything until we get to the end. You're going to see the color change yellow versus no color change. You can't see all these little molecules. You just see color change. Okay. All right, so here's what we got going on. First thing you're going to do in lab on Tuesday is you're going to take those HIV proteins. And you're going to put those HIV proteins, the HIV antigen, in every single well on your plate. You're going to let it sit for like 5 minutes. What's going to happen is these HIV proteins, these HIV antigens, stick to the well. First thing that has to happen. So every single well has the antigen, the HIV antigen. Next up, we add patient samples. Okay. Patient number 1 right here, you add their serum. Patient number 2, you add their serum. What's happening is you're trying to figure out if the patients have the antibodies against HIV. This patient, we can see the antibodies are attaching. This patient, we see the antibodies are not. These are not HIV antibodies. These are HIV antibodies. Right now, we can tell this first patient is positive, but we can't see that with the naked eye, which is why we have to do the rest of the steps. Okay, so we have them here. Now, it's very, very important after this step, okay, so you guys are going to add serum samples, let them sit for a while, give some time for them to absorb, then you're going to rinse them out. Rinsing is crucial because anything that is not attached needs to leave. These need to leave. If you don't get them all out, you're going to get a positive result. These, when you rinse, they're not going to come out because they are attached. Of course you're going to get a positive result. So next step, you rinse to remove unbound antibodies. You want to get rid of the ones that aren't attached. These don't come out because they're physically attached. These will because they're not. So again, positive result, negative result. We can't see this yet. So what do we have to do? What's called enzyme linked. Next step, we're going to add another antibody that has an enzyme. This antibody that has an enzyme on it will attach to any antibody present. It doesn't matter what it is, it's not specific. So it's attaching. Over here, who's it going to attach to? No No, buddy, because you rinsed them all out, which is why rinsing is crucial. If you didn't rinse them all out, this is going to attach, and then it's going to appear positive. Okay, so there's a lot of room for error, if you don't rinse properly. Okay, so this enzyme antibody attachments, enzyme linked. Enzyme substrate reactions. Remember, the substrate binds to the enzyme, reaction happens. So what's going to happen next is we need to provide a substrate. So you let this sit a while, okay? You'll rinse, so these enzymes leave. Okay, so we're going to rinse so they go away. These will not go away because they're attached. And then we add a substrate. We add a substrate. Substrate binds to the enzyme. Reaction happens. Turns yellow. Over here is a substrate, but there's no enzyme. Why is there no enzyme? Because there's no HIV antibody. What does that mean? It means the patient doesn't have HIV antibody, so they're negative. So because there's no enzyme, there's no color change. This one stays clear. This one, because substrate and enzyme reacted, gave off a color change, and the only reason that happened, because the HIV antibody is there, that bounds the HIV. It's a really cool test. A lot of them, of course, are automated, but we're going to do this by hand and see how it works. When we do it in lab, we're going to have brown versus clear for our results. We're going to see kind of a light chocolate brown color for our test results, but it just depends on, whatever test you're running, whatever types of substrates they use and enzymes. So that's the premise behind, that's the idea behind this type of test. Just kind of read up on those steps. This should make a little more sense to you now that, you've seen this. And then in vivo, once again, means within the body. The most common example is the tuberculin test. Okay, let's do our last concept check. So agglutination tests rely on blank, while precipitation tests rely on blank. So agglutination tests, what are we looking at?
Whole cell.
Whole cell. What about precipitation reactions? Soluble molecules. Yep. Western blot. That is used to confirm diagnosis of which viral infection? HIV. Very good. Indirect ELISA. What are we looking for in that patient's serum? Are we looking for antibodies or antigens?
Antibodies.
Antibodies. Yes. Indirect is for antibodies. Direct is for antigens.